Okay, so good morning, everyone. So we are going to start now with the live session with marine organisms. So you will have first Ilaria, then we will have Britta and Calli, and we have in the jury. So we have Sarah and Eric. So you are ready to start. As you know, you have 15 minutes. So we are live normally. I'm going to share the slides with you and we unmute. Okay. Whatever you want.
So good morning, everyone, and welcome to my Immersion project presentation on NLRC3-like function in Exaiptasia pallida innate immune response to a bacterial infection. I was supervised by François Seneca at the Centre Scientifique de Monaco, CSM. So the immune system is a complex process comprised of different molecular components that bind and protect an organism from pathogens. There are two different types of responses, the innate immune response and the adaptive immune response. The innate immune response immediately prevents the spread of the pathogen, and the adaptive immune response can perform specific attacks and can display memory. In invertebrates, both responses are present, whereas in invertebrates, only the innate immune response is present. So innate immunity presents a rich repertoire of receptors, which recognize and bind on the pathogen surface. This activates signaling cascades, which are followed by the induction of inflammation. Among these receptors, we have node-like receptors, NLRs, which are intracellular pattern recognition receptors. They are evolutionary conserved across plants and animals, and they can initiate and regulate many innate immune signaling pathways. They also have a role in the inflammasome in vertebrates. So there are 22 NLRs in humans that are known, but only 10 are well studied, whereas in mice, more than 30 are present. In fish and early divergent metazoans, such as cnidarians, NLRs are very rich and diverse, whereas in the main two models that are used to study innate immunity, such as the fruit fly Drosophila and the roundworm C. elegans, these receptors are absent. So a previous study at the Centre Scientifique de Monaco has shown that Exaiptasia pallida has a rich repertoire of NLRs and that these genes are regulated during infection. So here we will discuss about the homologous sequence of NLRC3, but why NLRC3? NLRC3 is the most recently characterized and least studied NLR. It is part of the regulatory subgroup with a CAR domain, and these NLRs can function as either positive or negative regulators of many signaling pathways. And in the case of NLRC3, it negatively regulates several inflammatory signaling pathways in both humans and fish. But what about the immune system in Exaiptasia pallida? Well, it's still unknown, but there are certain genes, such as NLRC3-like, that can have a potential role in immunity. So here we have a representation of the complexity of innate immunity pathways, as we can see, but we will focus on this pathway, so TLR2 and FKB pathway. So by having a close look to this pathway in human, we can see that we have a toll-like receptor, TLR, which detects the pathogen, and this activates the pathway, which has a cascade effect, and releases the NFKB and induces inflammation. When we compare this to Exaiptasia pallida, we know that there are certain members, such as TLR here, and the NEMO complex, that are not present in Exaiptasia. And also, we don't know the function of NLRC3-like, which in humans, it deubiquitinates a member of the pathway, and this deubiquitination leads to the inhibition of this pathway. The other way around. So the aim of this study is to investigate the function of NLRC3-like in Exaiptasia pallida innate immune response. And we would like to answer the question, how does NLRC3-like influence the TLR and FKB pathway? So our hypothesis is NLRC3-like negatively interacts with TRAF6. So in order to investigate the function of NLRC3-like, we used bioinformatic tools, such as UniProt-KB, which was used to retrieve sequences from other organisms that are similar to our protein of interest. In addition, conserved domain database was used to identify the different domains in NLRC3-like in order to prove that it is the homologous of the human NLRC3. And the local server at the CSM was used to retrieve sequences of Exaiptasia pallida and quickly blast them against other databases. So in order to do the alignment between the sequences, we use the software GelView Cluster Omega, and to create phylogenetic trees in order to see how the sequences are related to each other, we use the neighbor joining tool in GelView. In addition, we use the cell line HEK293 cell. These are human embryonic kidney cells, and they have been we've transfected them with 3 different plasmids encoding our protein of interest, NLRC3-like, plus a tag, which is GFP, HA, and MYC. But here in this presentation, we will be focusing on the first 2. The transfection was done with PI, and then after 48 hours of transfection, we ran Western blots. On the first day, the cells were lysed, the proteins were migrated on a 9% SDS page gel, then transferred onto a nitrocellulose membrane, and a primary antibody was applied on this overnight. And then on the next day, a secondary antibody was used, and the results were obtained. So in order to prove that NLRC3-like is homologous of NLRC3 in human, the NOT2 domain that was recently identified was compared to other NOT2 domain sequences from other organisms and human NLRC3. So here, as we can see in this tree, we can see how the NOT2 domain of NLRC3-like and the NOT2 domain of human NLRC3 are clustered together, meaning that they are closely related. So when we look at the structure of these 2 genes, we can see a similar domains between each other. And both of them have a NATCH domain, a NOT2 domain, and leucine-rich repeat domains. But NLRC3-like has a transmembrane domain, whereas NLRC3 is an intracellular gene, and it presents a CAR domain, which is not present in NLRC3-like. So these high similarities led to the 2 genes to get clustered together, and plus it proves that NLRC3-like is the homologous of NLRC3 in humans. So Schneider et al. Showed that NLRC3 in humans presents a TRAF binding motif. And this TRAF binding motif is responsible of the interaction between NLRC3 and TRAF6 in human. So this motif is comprised of different amino acids, as we can see here in the structure, and any combination of these amino acids can create a possible TRAF binding motif. So when we compare different sequences of different organisms and TRAF6 from Exaiptasia pallida, we can see how the TRAF binding sites are present in all the sequences, meaning that it is being conserved through evolution. And when we look at Exaiptasia pallida sequence, we can see, compared to the human one, that they have the same exact amino acids except for one. In Exaiptasia pallida, we have threonine, and in humans, we have serine. But a difference in one amino acid doesn't change the function of this site. And the chemical characteristics of these 2 compared sites do not differ because both serine and threonine are polar uncharged hydroxylic amino acids. So a possible interaction between NLRC3-like and TRAF6-like can potentially occur thanks to this binding site. Several studies have shown that TRAF6 in human has zinc fingers, which creates suitable site for inhibitors and or proteins to attach. And this site is known as hydrophobic packet. So in humans, the hydrophobic packet of TRAF6 is comprised of the and I don't remember the other name, 2 hydrophobic amino acids. I'm sorry for that. And here we have a list of hydrophobic amino acids, and any combination of these amino acids can create a possible hydrophobic packet in these sequences. So from the transcriptome of Exaiptasia pallida, we have different TRAF6-like candidates, and having an hydrophobic packet would make them the homologous of the TRAF6 of humans. So when we compared them, we saw that there are 3 candidates that can be the possible homologous of TRAF6 of humans, but one of them has exactly the same amino acids as the one in humans, but in a different order. The order, though, doesn't change the functionality of the site, as long as these amino acids are hydrophobic. And based on these results, there are three TRAF6-like candidates that can be possible homologous to TRAF6 in human. So the trough binding motif and the hydrophobic pocket could possibly be responsible of the interaction between NLRC3 and trough6 in human. So several studies, in fact, have shown that these binding sites are functionally relevant. In order to visualize the expression of our protein of interest, we use the tag green fluorescence protein, which allows us exactly to visualize this expression. So here we transfected the human cells with the plasmid encoding GFP empty, and over 60 hours of transfection, we saw that the cells expressed highly the plasmids, which results in a high fluorescence detection, as you can see, and meaning that the transfection has been successful. Whereas when the cells were transfected with the plasmid encoding GFP NLRC3, the expression appeared to be lower. In fact, we have a low fluorescence spread out in the cells. So we hypothesized that the size of the plasmid could have an effect on this expression level. In fact, when we compared the sizes of the two plasmids that I just mentioned, the GFP empty is smaller than GFP NLRC3. So we have a 30 kilodalton against 140 kilodalton. And as we saw, a smaller size had a better expression, and a bigger size had a more difficult expression. And also, HA NLRC3 has a smaller size compared to GFP NLRC3. And in fact, it presented a high protein abundance and expression, which we detected through the clear and defined bands in the Western blots. So here we tested different concentrations of plasmids to better see, to understand which concentration was the best for protein expression. So as we can see here for HA NLRC3, we have the concentrations going from 50 nanograms to 2000 nanograms, and the best concentration appears to be 500 nanograms because we have a clear and defined band, whereas the band starts to be less unclear when the concentration increases, and it's not present in lower concentrations. We have a similar pattern when we look at the GFP NLRC3 results. In fact, we have 500 nanograms, again, to be the best concentration to use. So over 500 nanogram, we saw in both scenarios that the expression level was lower. And we hypothesized that a higher plasmid concentration could have a toxic effect in the cells. Below 500 nanograms, the expression level was again lower. So we hypothesized that a lower plasmid concentration could have not enough protein for the cells to express. So in conclusion, NLRC3-like and NLRC3 from human are closely related and could potentially have a similar function. And this suggests that these genes have been conserved through evolution, and that early divergent organisms, such as cnidarians, can be used to better understand the immune system in humans. NLRC3-like and trough6-like could potentially interact, thanks to the trough binding motif and the hydrophobic pocket that we identified. So this is a first functional study on NLRs in cnidarians. So future studies are required in order to better understand the function of NLRC3-like with the TLR NFKB pathway. And these studies could use experiments such as co-immunoprecipitation, co-transfection, and siRNA to better understand this interaction. So thank you very much for your attention, and I would like to thank my supervisor, Francois Seneca, that helped me throughout the internship and shared with me all his knowledge about this topic.
So thank you, Ilaria. I think we're going to open the question. Do you want first to have the student, or do you want to do? Okay.
What was your favorite part of the methodology? As I know this was the first time that you've been working with in situ hybridization and the analysis of genes.
Mm-hmm.
What was, because the Western blots, I know you had some.
I like yes, but I like Western blots because they were very challenging for me. So I had some problems at the beginning, but I think that was the best method that I used, I would say, and also cell growth. Yeah.
Thank you.
No, thank you.
Any other questions?
Thank you. Thank you very much for the presentation.
I have a first general question. You said that you presented this presentation results from a specific plasmid, but you worked also on other.
Yes.
can you just mention a little bit what is the effect of plasmid and if there is a change?
Yes. So as I mentioned, I use also the MIC HA and LRC3, which I didn't present because it wasn't really successful when we did Western blots. So we didn't have clear results when we used this plasmid. So it was a bit tricky for us to work with it. So the best one that we had working was HA compared to all the other ones, and then GFP. Also, we tried flag trap sticks, so another plasmid. And here again, we had some complicances, so some challenges. So
Remember just what is the different role of these different plasmid? Can you explain why it's important
to try different ones? It depends. So how I understand, from what I understood, is that each tag has a different antibody. So maybe this can have a different effect on the results. So maybe some can be more detected than the other ones, and some maybe are just easier to express, as I said, also because of the size, probably, what we hypothesize. But yes, I don't know if this answers really your question, but yeah, sure. Okay.
I can continue. Thank you very much. Very interesting. A lot of questions. I will start by your tree.
Yes.
Phylogenetic tree. And I didn't really get. Here we go. You say that the node 2 domain clusters with the human one.
Yes.
But you have plenty of other cnidarians in here as well. I think so. Especially if you use the CSM database. I know they have tons of corals and cnidarian genomes or sequences in there.
Here, it's not specifically on cnidarians. Sorry for interrupting.
Okay, so because I'm very surprised to see that the human and the Exaiptasia node 2 are clustered together, especially if in the next slide. Afterwards. Here we go. I see a node 2 domain in Exaiptasia, but not in the human one.
It's written here because they are together in the NDB domain. So I put them together in the representation, even though probably I should have divided them.
Okay.
Sorry for the visualization.
But still, I'm still very surprised that it's so closely related. And then
So,
what are the outgroups that you used? And yeah.
So this is mainly done, if I have to be completely transparent, by my supervisor.
Okay.
So as far as I understood, the node 2 domains from different organisms, such as they're not NLRC3 sequences on this side here, but like the organisms are very different. So we have mice, we have cows, we have many different ones, even chicken, for example. So it was, what I assume was that he really focused on the node 2 domain present in many different organisms and saw how this could be related to each other with NLRC3-like.
Okay, but that's on the node 2. So it tells us something about node 2, but not necessarily on the NLR family. So it can be sort of dissociated.
Yeah, because yes, this was done because we just identified this domain. So that's why this has been done, but I understand what you're saying. Yeah.
Or perhaps some other analysis of the NLR family. The other questions, why did you use the hex cells? So you said it's a kidney.
Yes.
So why did you do, why this kind of cells?
We use them because they're very easy to grow, and plus they are very resistant to transfection. And they are also very successful in expressing the plasmids. This is the main reason. Because we tried also other cells.
Okay, but what is the question now? What does the transfection do? So you want to show that the Exaiptasia cells are expressed in mammalian cells? Or
No.
Because, can you explain a little bit?
Of course,
what
because we don't have enough tools to actually investigate the proteins from these animals. So we have to use human cells in order to actually study them. So the human cells are used as a tool to just reproduce the protein. And here, this is like a first, how can I say it? it's really the beginning of it. So we had to make sure that the protein from the anemone is expressed, so that then for the next steps we can express as well other proteins and really visualize how if the two are interacting. So with immunoprecipitation and with co-immunoprecipitation mainly.
So because the idea behind this was to transfect the NLR and the trough
Correct.
and see if it blocks.
So if the two are interacting, we will see that they are linked together. So one of them is gonna have a tag and the other one another one. So let's say we have HA and LRC3 and flag TRAF6. So let's say they interact and they are in the solution together. Then with co-immunoprecipitation, we link NLRC3, for example, with an antibody with a bead. And then this allows the precipitation of the protein when we spin this solution. And then we remove the other proteins. Then we wash and everything. And then when you go to detect TRAF6, for example, so you know that you didn't use a primary antibody for TRAF6. So if it's there, you're going to detect it, but it means because it's interacting with the other one. So you go and try to find flag, the tag. And then if you see a band for TRAF6, it means that it's interacting with HA and LRC3, and then vice versa as well. I don't know if it's the answer.
It makes total sense. My question here also is, can you activate the TLR pathway in those head cells and then see if the Exaiptasia gene can block the whole pathway? Because that's a prediction, right?
Yes.
Independently of interacting with another Exaiptasia gene, but it could potentially also block the TRAF6 of the human.
That's a good question. I'm not very sure about it, but I can get back to you. I will discuss about this with Francois, because I remember he explained to me, because I had the same question, but I really don't remember. I'm sorry.
Fair enough.
Yeah. Oh yeah, we start by.
I didn't 100% get what you meant when you talked about the kilodalton difference and the transfection efficiency.
So I can go back probably to the slide. So it's an hypothesis. I don't understand this. It's an hypothesis that we made because we were continuously seeing that when we transfected the cells with GFP, just the tag, we had a very high expression. And also with HA, we had better results compared to GFP, which has the highest size. So what we hypothesized was that the size could have an effect on the expression. So let's say you put something very big in the cells and they would struggle more to reproduce it. So that's the idea, what we thought.
Okay. So here you're showing the protein size of the tagged NLRC, not of the plasmid, right? The size, because it's a plasmid size that is a problem of
this is just
the protein once it's translated.
Yes, sorry, this is just the protein plus the GFP. So it's what the cells are expressing.
Okay.
So,
and that's why I was confused, because it was more the plasmid size.
No, it's just the protein with the tag.
Okay, but then also you mentioned also it might have some toxicity effects, right?
Yes, correct.
It may have another explanation.
Exactly. This is again an assumption that we made because, yes, based on the Western blots here. Oh my gosh, here. Because we were always seeing a nice clear band at that concentration, whereas in certain Western blots we didn't have any bands here, like at all. So we thought probably there are not enough proteins, because I mean, if there is enough proteins, you're gonna detect it. And also when at the beginning I didn't know that I had to resuspend the solution, I didn't get many clear bands. So that was another thought that we had, because maybe not having enough proteins didn't show a clear band.
And again, here you look at the effectively translated protein and not on the amount of plasmid that was transfected. So do you know how you could check if you efficiently transfected the plasmid?
But like for this specific plasmid, or so for GFP and LRC3, for example, or
For this plasmid, yes.
well, for
whatever
GFP,
plasmid, yeah.
it's quite simple because we can detect it with the fluorescence.
Right, but that's a protein. It's already
Yes.
translated.
Yes.
So if it's a problem of transfection because of the size of the plasmid, it might be linked, but or it's a toxicity problem once you have an overexpression of your NLR. The DNA level of the plasmid is something different of the protein level that is translated once the plasmid has been transfected.
Yes,
Know what
I
mean?
understand. Yes, I don't know. I really I have. I'm not sure I know about this.
Well, it's by PCR, by qPCR.
Okay.
You can measure sort of
Okay.
the amount of DNA that has been transfected. And I have one last thing where I would like to tease you a little bit on your conclusion.
Okay.
If you go to the slide. You say that it helps to get a better understanding of the human immune system.
Mhm.
Can you develop that a little bit further, especially knowing that you had to use human cells to test your Exaiptasia genes?
Okay. Let me think 1 second. No, so because there are, as you know, different models that have been used, such as Drosophila and the C. elegans, and they showed that there are that they don't show certain genes that are found in other early divergent organisms. So this is why I said this, because you can use anemones, for example, to better understand this immune system, because they present really a high number of genes that are very similar to humans. And this is the first part of the answer. Second part, wait, I forgot
I know
already.
I'm
Like
teasing
the
you.
No, it's
you're
okay.
not confronted to this kind of question.
But I know that we use human cells, but because we really don't have any other tools right now to investigate this. So this is like what
But still, to push it a little bit further. So
Yeah.
why do you think that you would learn more about the human immune system using the Exaiptasia gene rather than using the human gene that is understudied, as you said?
Because how I see it, Exaiptasia doesn't have an adaptive immune system. So in humans, you have the adaptive immune system that could interfere with the innate immune system. So that's why we use organisms that just have the innate immune system.
Okay.
This is what I.
Fair enough. Then I made one last question. Do you know in Drosophila and C. elegans, where those genes are lacking, how the innate immune system is controlled, if not by this large family of genes? Might be a little bit off.
Yes. I think, if I'm not wrong, they have TLRs, but they don't present NLRs. So this is how far I went into this. I know a little bit about other anemones and corals, so I was more focused on cnidaries. Yeah.
I'm always wondering about the C. elegans and Drosophila because they're lacking some key genes.
They do.
The NLRs are important to control.
They need to, yeah.
So is it always on? Anyway, I will look into this. Okay, I'm good.
Okay.
I don't know how your time was, but
No, I think we're good, yeah, about it. I want you to look at the screen because there is like some people on YouTube
Thank you. Thanks.
saying like, brava,
Francois?
from Ocean, and I get that.
Thank you.
So thank you again,
Thank you.
Ilaria.
Thank you very much.
And we are going to take like 15 minutes break, and we are starting at 10:15 for you, Britta. Okay? Okay.
Okay.
Okay, so
Hello, world.
yes, we are back live. As you can see, Britta is going to start. We will have just the Calli after. So ready to start, Britta? Yes?
Yes, on time. I'm gonna set the timer for myself
Yes,
as well.
so you have 15 minutes, as you know, with 15 minutes of question. You can ask question, guys.
Are you ready?
So we have the slides. You can start whenever you want. Yeah.
Okay, perfect. So, good morning and welcome to the third presentation of the day, where I'm going to present to you the work I conducted on population genetics of Red Sea coral gobies. I was supervised by Dr. Song He at the Reef Ecology Lab in Kaus, Saudi Arabia. But before I dive into the work I did, I would like to give you a brief introduction on the Red Sea in general. So, the Red Sea is a very important biodiversity hotspot with a high rate of endemism of about 14%. Now, this high rate can be explained by the unique geographical conditions of the Red Sea. So while it used to be directly connected to the Indian Ocean, the water levels dropped significantly to 130 meters during the Pleistocene ice ages, and the water exchange is limited ever since. Now, the Red Sea is partially isolated through the Bab el-Mandab Strait in the south here. Also, the Red Sea is characterized by several environmental gradients, which stretch along the main axis of the Red Sea. And there's a marked environmental shift at about 20 degrees latitude. So these environmental gradients significantly influence the biological communities in the Red Sea. What's also interesting is that the Red Sea is heavily understudied. A study by Mariana et al. In 2013 found that there's only 1/6 of research compared to the Great Barrier Reef, and only 1/8 compared to the Caribbean. So there's a huge knowledge gap that the Reef Ecology Lab aims to fill in. And this is why in April this year, they launched a new project, Population Genetics of Coral Gobies in the Red Sea, which is part of a bigger framework. So since this is a big undertaking, I was part of it in its early stages, and it's still ongoing. So this is why today I'm going to present to you the work I conducted while I was there. So why do we look at our cryptobenthic fish? So while there is a huge knowledge gap for the Red Sea, this is especially relevant within cryptic species. So cryptobenthic fish are estimated to account for about 40% of all non-reef fishes, and yet very little is known about their ecology, biology, and evolution. One of these understudied cryptobenthic fish are gobidon. Currently, there are 28 species described for the whole Indo-Pacific region, and 9 of them being described in the Red Sea. These coral-dwelling fish have a mutualistic relationship with acropora corals, as they seek shelter from predators in the physical structures. Coral gobies, or gobidon, are very host-specialized fish. They have high host fidelity, little post-settlement migration. Also, these fish lack scales and have very species-specific life stage coloration. Here I put up an example for you, which is not 4 different species, but 1. So this is Gobidon bilineatus, with 2 adults up here, and then we have a subadult and a juvenile here. So you can see the identification is challenging. But why do we look at population genetics in the first place? So population genetics is a very useful tool for studying the microevolution, population dynamics, and it's also highly used in conservation genetics. The aim of this project is to assess the connectivity between the different populations, and this will first contribute significantly to the general understanding of gene flow patterns and the biodiversity in the Red Sea. Secondly, it also fills in the knowledge gap for these understudied cryptobenthic fish. So the research question is, what is the genetic distance between the different populations? And our hypothesis is based on the limited dispersal range of the fish and the high host fidelity. So hence we hypothesize that there are multiple populations of gobidon in the Red Sea. Moving on to the methodology. So here you see a map of the different sampling locations. The red points indicate the places that I sampled. So Kaust is marked here, which is in the center. Here we sampled mostly in inshore reefs. And then there were 2 different research expeditions that took place. The first one going south to Alif and the Farazan banks, which is comprised of a group of offshore islands. And then the second one went up north to Rabik and Yanbu. So before I collected any fish, I conducted a training in animal ethics and handling, and the whole research was approved by CAUS's Biosafety and Animal Ethics Committee. So for the fish collections, I have a video that unfortunately I cannot share with you on the slides. I'm just going to show it to you here. So I sampled the fish scuba diving, and I anesthetized the fish using clove oil diluted in ethanol and seawater, and then collected them in falcon tubes. Then at the surface, I took data on the sampling date, the GPS location, the exposure of the reef, the depth if possible, and some other factors. And a CAUS colleague helped me with the morphological identification of some species if possible. Then, after the fish were collected, I extracted their DNA using the D&EZ blood and tissue kit, following the manufacturer's instructions. The samples were incubated until the tissue was fully lysed and eluted once in the last step. Then afterwards, it's really important to measure the DNA yield to make sure that the extraction was done successfully. And I used this using the qubit fluorometer, which gives highly accurate results and quantifies the DNA, and then the results are in nanograms per microliter. Later, the extracted DNA was stored at -20 degrees Celsius. So these are preliminary results of what I have done up to now. So 129 gobidon were collected in total, and I covered a total distance of about 600 kilometers. So this table gives a very brief first overview of what I did. You have the different regions of the Red Sea and the collected fish that I found here, as well as the start and the end date of the sampling trips and the minimum yield that I got for each group. The average sampling depth was 6.4 meters, and about 95% of all fish were found at sheltered sides of the reef. So let's have a look at the gobidon that could be identified. So out of the 129 specimens, 25 individuals in 6 taxa could be identified. So here you have the bar plot of the different species, and A correspond to not identified. And the photos up here are in the same order as the plots here. So we have Gobidon bilinearis here on the far left. So what you can see here, that Gobidon irregularis, this one here, had the highest counts with 10 species, followed by Gobidon bilinearis with 7. But what about the different sampling locations? So as you can see here, most fish were collected at Jeddah, which is very, very close to CAUS here on the map. And then it's followed by the Farazan banks. The highest distribution of identified fish are here in the Farazan banks in the far south, where we have 5 irregularis and 5 bilinearis, which are the most identified and most sampled species that I got. So what we can also see here, that there's an uneven distribution across the sampling locations. So the first sampling trip to the south, Alif and the Farazan banks, here I collected 46 specimens, whereas for the northern research cruise to Rabik, Yanbu South, I only collected 38, whereas sampling efforts were comparable. So what could explain this uneven distribution across the locations? So we do know that gobidon is dependent on acropora presence, but also current and exposure plays a role. So actually, when we sampled in the north, we had a lot more exposed sites where acropora was generally present, but the gobidon seemed to avoid stronger currents by hiding in the coral crevices, and hence I couldn't find them there. Also, we had a few more wall dives in the north, which is not the preferred habitat for acropora. So the next thing I want to discuss is the species identification, which is highly difficult. I tried to study the morphological differences, but I realized that it just takes experience and training, which I didn't have. So I couldn't identify the species with confidence. But even the goby expert in CAUS often had difficulties distinguishing between these two species here, which is Irregularis here on the right and Bilinearis on the left. We can discuss that later if you want. Good, moving on to the sampling size. So 129 gobidon in total are not much. And then even if we break it down to the different species we got, it will be even lower. So other studies had a lot higher sampling size of maybe 550 fish, or one study I found even had almost 1,000 samples. However, I found a study by Halle et al. That found that even a small size of 25 to 30 samples is sufficient for a microsatellite-based population genetic study. So based on the assumption that most of the fish I collected belong to the two species Irregularis and Billiniaris, then we can assume that it's probably big enough to conduct the analysis based on this hypothesis. So there's still a lot of work that has to be conducted in order to obtain results for a full population analysis. So what are the next steps? First, after the extraction, we will have to conduct PCRs, and then it's usually a difficulty to find the right primers that amplify the targeted gene. Then after amplification, the gene will have to be sequenced, and then through DNA barcoding, we can molecularly identify the species. In the successive step, the genetic distance will have to be calculated and analyzed, and then in the last step, the phylogeographic distances will have to be visualized, which is typically done using haplotype networks, which can look like this. So since there are very, very few, I couldn't find any study on cryptobenthic fish in the Red Sea, I had to look at other regions. So this is an analog study that I used that was conducted on the cryptobenthic 3-fin blenny in the Adriatic Sea. What you can see in this haplotype network here is that the colors correspond to the different sampling locations. And while the northern populations in blue are more well mixed, we do see a break for the southern one, and here for the central and southern Adriatic Sea, it's really mixed because the populations share the same haplotype. So what's interesting is in this study, they actually identified the circulation systems to be the major driver for structuring populations. So based on this, maybe we have to rephrase our research hypothesis, but there are some other factors we should look into. For example, depth. So depth has been identified to be one of these major forces, and the forces that structure populations are stronger at a shallow depth. So since our goby don't live very shallow, this would actually favor our initial hypothesis that there are multiple populations. But there are many, many more environmental conditions that have to be considered, such as the environmental gradients that we have in the Red Sea. So here I looked at a study on an anemone fish, which was conducted in the Red Sea, which has a very short pelagic larval duration of less than 2 weeks and a very, very shallow depth. And yet they came to the same conclusion as in the previous study, that it's actually the surface circulation, which is the key driver for structuring the populations. So in this study, they found a well-mixed population with a genetic break at a latitude of 19 degrees north. There are a few limitations to the study. So firstly, it's not finished yet, and it's still ongoing. It was definitely the plan that I finish all the steps, including the analysis, but this was not possible due to maybe underestimation of the whole workload and some other factors. Secondly, we couldn't sample in all the sites that we aimed to. So the red ones again correspond to the sites that I actually visited, and the green ones up here are the planned ones. So we couldn't go here because the necessary research permit wasn't obtained. But in order to conduct a full analysis covering the whole geographic range of the Red Sea, it is crucial to also have samples from the far north. So if it's not possible to sample in Saudi Arabia, then maybe trips to Egypt could be organized instead. Also in this study, we only focused on the eastern shoreline of the Red Sea, but it would also be very interesting to have samples along the western shoreline of the Red Sea. Then we would be able to capture the whole process of the gene flow and maybe identify patterns for an east-west connectivity in the Red Sea. Thirdly, I think it would be a good practice to establish a baseline survey of general gobidon distribution and abundance. Because according to the IUCN Red List, many gobidon species are listed as vulnerable, and their host acropora are near threatened due to climate change. And since we know that these fish are very host specialized, this is especially dramatic, and which is why this information would not only provide more guidance for this specific study, but it would also contribute to the general understanding of these small cryptobenthic fish. So to conclude on this study, 25 out of the 129 gobidon could be identified, with Gobidon irregularis being the most abundant. Secondly, surface circulation has shown to be the major driver as opposed to maybe a limited dispersal range or geographic distance. Thirdly, the study on an anemone fish by Mariana et al. Found a genetic break at a latitude of 19 degrees north. And if you remember in the beginning, when I introduced the environmental gradients, there is the marked environmental shift at 20 degrees north, which was identified by other studies. So based on this, we can assume that each species might have a slightly different genetic gradient across the Red Sea. And secondly, we know now that there are several mechanisms that cause the genetic differentiation between species. Populations, sorry. So this is why the research hypothesis should be rephrased, and I suggest to do it as follows. So the Gobidon sampled south of the approximately 19 degrees north genetic break will show a higher genetic variation compared to the samples in the north. And the continuation of this study is very important as it will contribute significantly to the understanding of general gene flow patterns in the Red Sea. And also it will give information on the connectivity of a cryptobenthic fish. So there are many, many people that I would like to thank, most importantly the Reef Ecology Lab, because I was able to learn so many things there, and I was involved in many, many other research projects than just this one. Also amazing sampling support and people that taught me lab skills. And also I would like to thank you for your attention, and I'm excited to hear questions.
Any questions, guys? Do you want to start? Yes.
What was your favorite area to sample of all the sites?
My
Like, what had the
favorite,
most, like, maybe, I don't know, it's hard to say like biodiversity, but like, what was the most lively site? Or like your favorite?
for fish or for diving?
As you feel, as you feel.
I really, really enjoyed, I'm going to show you the map again, the Farah sandbanks in the far south. It was absolutely amazing. The water temperature, I mean, it's not very great for the corals, but it was very comfortable water temperature. And also we saw loads of sharks there, so it was amazing. I was a little unhappy with that, but the other people that went with me on the cruise saw many hammerhead sharks. But I was always very shallow looking at my gobies, so I missed what's going on behind my back. Yeah.
Yes, Mariana.
Yeah, for the papers you said that you need a permit to go sampling.
Yeah.
It's the same country, so do you think it's a little bit
Yes,
harder
so
to get it from
here we have a specific problem due to time restrictions. I couldn't mention it, but actually this area here is very special. So Saudi Arabia is building some massive giga projects, and one of these projects is a huge city that they're building just in this area here. So this city is called Neom, and by 2045 they aim to have 9 million people living there. But it's still a construction site, so it's very interesting looking at the visions that Saudi Arabia has, because they're ambitious. They're very ambitious. But yeah, this is the reason, because the research permit had to be obtained from the Neom, from this mega city administration office. So that's why I think Egypt is maybe the better option.
Okay. That's crazy.
It is crazy.
Yeah, with all the water they have, it's wild.
As well as I have a connected question. So you mentioned at the beginning that the Red Sea is understudied.
Yes.
Do you know the reasons for that?
The reasons can be easily explained looking at the political situations of the bordering countries. So I don't know when research has started in the Great Barrier Reef, but I assume in the 20th century, whereas the Red Sea had been restricted to the public for a very, very long time. And yeah, which is why in recent years, especially Saudi Arabia has invested massively into contributing to an understanding of the Red Sea and to, well, fill up this knowledge gap.
It connects to your perspective where you say it would be interesting to see the other parts of the Red Sea for sampling. It might be tricky to get permits.
So I know Egypt is usually not a problem, as I was told. Sudan is also possible, also great for shark diving. And Eritrea, I don't know. Yemen, you can forget about it, especially if you aim to go to Yemen from Saudi Arabia. But at least Egypt and Sudan should be possible to organize. Hello, Paola.
Hello. I have you. Sorry for the delay, but I was out of order this morning. Now it's better.
No worries.
May I ask you some questions?
Yes,
There
of
is
course.
some people in the audience.
I think you can go
Okay.
ahead.
Okay, so I have some questions. First of all, on your DNA yield.
Yes.
I saw that there is a relationship between the location and the DNA yield. So I would like if you can explain this is just casual or there is a reason behind the fact that you have quite a factor 10 between the central, for example, and the north DNA capacity yield.
I didn't look into that statistically, but I didn't extract my samples in a chronological order. So I probably extracted samples from the southern region and the northern at the same time. So I would assume that this is completely random. The
Okay,
minimum.
so if
Sorry, no, go ahead.
no, but it's perfect if it's random. So it's again highlight that there is some difference in this DNA yield. And so could be to the species or to the locations, or I don't know if you have some idea about that. No,
Yeah,
if it's random, it's not yourself.
I don't know. I mean, sadly, I couldn't amplify the DNA, so we don't know if my DNA is in there, but I hope not.
I have a sort of a connected question. So here, why did you choose the minimum DNA here? Because 0.40 nanograms per microliter is not much, and you probably can't do any further experiments with this because it's so low. But why didn't you take the average DNA yield to get an idea of?
So the average DNA yield is about 12, and the highest range was 46 point something nanograms per microliter. So I looked into the literature, and while these yields seem very low, and for human DNA, for example, they prefer 5 nanograms per microliter as the minimum threshold. However, for fish, the yield really depends on the body part where you extract the DNA from. So I took fin clips of the right pectoral fin, which is known to give lower yields as opposed to maybe taking it from the liver or from the gills of the fish. So I looked into the literature, and studies found that worked on the DNA extraction from fish from different body parts. They said that their minimum threshold is 0.05, which is very, very low. So I guess it's a trial and error. Like, this is a very, very low yield of 0.14. So I assume for this sample, if I don't manage to amplify DNA, I would just redo the extractions. But yeah, I was assuming the minimum threshold of 0.05 nanograms per microliter.
I have a follow-up question. So you said that you were using the tail for the DNA extraction, but if I got it right, you sampled
the pectoral fin.
Oh, sorry.
Yeah.
But you sampled the whole fish for it.
Yes. So the fish sampling is very tricky because the fish are very fast and they really hide in the coral crevices. So it's not really feasible to anesthetize and take the clip and release them back
But you sampled them by anesthetizing them with the cloth oil.
Yeah, but they were still moving, so I collected the whole fish as I was taught by.
Okay, I think you know where I'm getting at, because normally, well, often in fish studies, if they don't look at other organs as well, they just use the tail or whatever, and then release the fish, because sometimes fish can regenerate some of their appendages.
Yeah.
So especially if you're looking at threatened species or something, it's probably.
It's true. And also the study on the anemone fish that I mentioned, they actually just took a fin clip. They didn't collect the whole 991 fish that they sampled. So it would actually be interesting to go back into the water and try to take a fin clip without having to kill the fish. What's also interesting, what I found in the literature, is that environmental eDNA might be a very promising solution in the future, but it's still in its infancy of what I read. But it has, I think, huge potentials also for population genetics.
Thank you very much, Britta, for the presentation. I have some questions. First of all, starting from your conclusion.
Yes.
In your conclusion, you mentioned that circulation is one of the major drivers. Is it something that you find from your results, or are you assuming it due to the literature?
I assumed this based on the literature, because when we phrased our original hypothesis, this was really just based on the huge geographic range and limited dispersal range. And benthic fish usually have a shorter pelagic larval duration, than other fish. But I couldn't find clear numbers for Gobidon. So for other species, the PLD ranges from 13 to 51, but for Gobidon, we don't have much information. So which is why I looked at other studies to identify patterns like, okay, what is it that shapes populations? And well, yeah, several studies have shown that it's circulation.
Do you know a little bit the circulation in the Red Sea?
I looked at patterns, so it's not very well understood compared to other systems. And this is mostly because due to a lack of in situ data. So which is why other studies then used satellite data, remote sensing, and they also used dispersal models. But even these studies argue that there's just not enough information, that there needs to be more in situ data to, well, have reliable information and, well, to have clear conclusions on that.
Okay, thank you. I have another question, just a clarification. So in your study, you work on gobies.
Yes.
Specifically, okay, and you say that for the moment, 9 species of gobies has been identified in the Red Sea.
6, yes.
28. And in your study, thanks to the help with the expert, you identify 6 species.
Ah, that's what you mean. Yes, yes.
And so what about the other non-identified individuals? Do you have some information that allow you to understand if these are in the 3 species that are known in the Red Sea, or it might be also other species that have never been observed? I don't know if you already have some information.
I have to admit, I didn't really look into that too much, but what I can say is that the last research that has been conducted on Gobidon in the Red Sea is from 2013 by this man, Jürgen Herrler. So in 2013 was really the last paper that was found on Gobidon for the Red Sea, where he identified 4 new species, including Irregularis and Bilineatis, which had the highest counts in my study here. So there's actually a high chance that in my samples there are unidentified species and there could be new ones described. It could also be that there are the remaining 3 ones that were described in the Red Sea in this sample size.
Yeah. The expert with who you worked in didn't provide some information that you don't have an idea of.
No, so he's the goby expert, but he was working on a different project. So I was showing him the samples that I had, but he wasn't involved too much in the whole process because he's more the taxonomy guy. But no, it's right. It would make sense to collaborate more, especially if we do have the know-how in KAUST. And yeah, and to involve the people that are actually knowledgeable about these fish.
We have a little bit time for
1 minute.
okay, so
Last question.
my question is very, very global. So what is the occurrence of those Gobidon in the corals, and is the risk of mortality for the coral higher if the Gobidon is absent, or is anything known about this?
I don't know. I didn't look into that, but what I found is that after climate-driven disturbances, for example, such as bleaching for the corals, the fish do not recover as fast as the host coral. So the fish will likely go extinct sooner than the actual coral. We don't know that. Maybe, maybe not.
Why that? Because he needs the microalgae of the coral to survive?
Now I have to think. So the study that I found that looked into the recovery rate of coral gobies after the disturbances, they said that the gobies were then, after this disturbance, not found in the coral at all. And some gobies then returned to their original host coral later, but not all of them. But I have to look into the study again, if I'm honest.
Okay, I'm good. Thank you.
Okay, so we are now with the presentation of Calli, which is going to start. So yeah, like Eric said, Paola, I'll just leave you in the back. I guess you can listen to the presentation of Calli, but you will say a word perhaps at the end. If you're ready, Calli? Yeah, you have the time with you? So ready for like 15 minutes?
Yeah, and then
Slides.
the slides are on the, do you mind? I'm just gonna, okay, perfect.
Perfect. Okay, let's start.
All right, so hello everyone, and thank you for joining me for my presentation regarding the Terra Pacific expedition and the coral health assessments that I conducted on coral samples from the Great Barrier Reef, Japan, and Fiji. So why did we conduct a study on coral reefs? Why do we care about the health of coral reefs? So essentially, as we touched on a little bit previously with Britta, coral reefs serve as a biodiversity hotspot for roughly 30% of the marine species in the oceans, though they cover very little surface area in proportion. So essentially, coral reefs are extremely important for ecosystem structure and function, as they provide biodiversity, which ultimately results in the increase of productivity among ecosystems and allows ecosystems to better cope with stresses that may be subjected to the reefs. In addition, there are anthropogenic interests of coral reefs, and in the United States alone, $3.4 billion US dollars is what is the yearly value for coral reefs. In addition, their biodiversity and ecosystems provide food resources for humans, as well as supports fishing activities. And in addition, they provide coastal protection through wave mitigation. They are also been recently used for medicinal research, as they can be extracted for their compounds that have recently been linked to possible solutions in cancer research. So as we can see, there's many significant value within coral reefs. Though coral reefs exhibit high economic and ecosystem values, they also face high levels of stress and anthropogenic threats. So some of the anthropogenic threats, just to name a few, are going to be pollution, overfishing, and destructive fishing, including the use of cyanide and dynamite, which can be extremely detrimental for reefs. So as we can see on this giant graph, this is from the IPCC report of 2021, and it depicts the projected temperature increase while facing rapid climate change. So the best case scenario is depicted in blue, though it still results in a 1 to 2 degree increase in global temperatures. However, the worst case scenario depicts a 4 to 5 degree increase. Regardless of the scenario, best or worst case, we still have an issue for our coral reefs as they exist in a specific ecological niche that allows for their physiology to properly function with low variance of temperature. So it has also been proven in the literature that high temperature increases with in the environments of coral reefs results in a reduction of symbionts. So what are symbionts and how do we measure like whether a coral is healthy or not? I'm going to use the whiteboard for those who are online. I'll try to highlight what I'm writing. So essentially we can assume, and in this study particularly, the corals exist in symbiosis. What this means is that there's photosynthetic algae that exists within the corals that provides sugars and lipids that will ultimately result in the increase of animal tissue biomass. So let's pretend this is a zooxanthella, which is also known as is another name for the symbiotic algae. And so these are our two energy producers of the cell. We'll just do a really quick mitochondria. This is very rough, so don't judge my drawings. This is a chloroplast, for example, photosystem 2. When we have external stressors such as increase in temperature, what the cells do is produce an increased amount of reactive oxygen species, which essentially in too high amounts can be damaging to the cell's DNA, RNA, and can ultimately result in cell death. So one of the mitigations of the cell that exists are antioxidant response. And essentially what it does is it binds here as our antioxidant, very clever name, and it deactivates the reactive oxygen species. So let's say that we have a cell that doesn't have a lot of antioxidant capacity. We have our functional proteins that we need for proper cell function. When we have them, they're usually in these tight polypeptide corals. When we have reactive oxygen species within the cell, we have an additional oxygen that binds to the carbon, which denatures the structure of the protein, essentially making it unusable to the cell. It loses its function. When this happens, based on the literature, we know this as carbonylation. When carbonylation occurs, the cell has another way of trying to mitigate this potential damage, and it is covered with ubiquitin ligases. And essentially what this does is it flags the cell for degradation through protosomal degradation. So here we have 1, 2, 3 different ways of ultimately determining whether a coral is expressing an unhealthy phenotype at the time of sampling. So in context, when we have an increase of carbonylation, antioxidant capacity, and ubiquitination, it could be an indication of bleaching, mortality, and susceptibility. So these are going to be the antioxidant states. These are going to represent the trophic states, which is related to the nutrient content of the corals. So as I previously mentioned, when we have photosynthesis, we have we have sugars and lipids that are produced, ultimately feeding the coral host animal, resulting in higher biomass. So when we have a reduction, it could also indicate that we're in an unhealthy status. So why does this matter? Why do we care about coral health? What is Terra Pacific? So Terra Pacific is actually the largest coral sampling effort that has ever been conducted in the Pacific Ocean. It's of extreme importance. As we all know, we are facing climate change and uncharted territories for the future for our ecosystem and for the function of extremely susceptible organisms, such as corals who exist in specific ecological niche. So the Terra Pacific left from Lorient, France in 2016, and on this blue is going to be the outbound trip. The orange is going to be the inbound trip of 1 year. The purpose of this was to collect as much data as possible. It's also important to note that the corals that were sampled and included in this study were sampled from unbleached reefs. So for my specific participation in this study, I had 4 islands assessed. So the 2 islands in the northern part are going to be 2 Japanese islands, Sissoko Island and Ogasawara. These islands represent temperate environments, meaning that there is high variation within the temperature throughout the year. Further assumptions can be made about the nutrients as we have thermocline mixing for nutrient availability. Then in the southern tropical environments that are defined by less variance of temperatures throughout the years, so we have less nutrient availability due to a decrease of water mixing. we have our islands, the Great Barrier Reef and Fiji. So the ultimate consideration of my individual study and the question that I aim to answer is, are there any significant differences between reefs in temperate versus tropical environments? So to answer this question, how are we going to do that? So we're going to use the 2 coral species that were sampled throughout the Terra Pacific expedition. The significance of these 2 coral species is that they are both emblematic of the Pacific Ocean, meaning that they're present in all of the locations, meaning they make really good replicates for not only the sampling efforts, but the methodology as well. In addition, they are reef-building corals, so they're super important for ecosystem function, and they serve as keystone species. The methodology, as touched on a little bit, included the trophic states and the oxidative states of the corals to make contextual inferences indicating the coral health assessment at the time of sampling. It's important to note that these are temporal samples, meaning that they exist at it's like a screenshot in time of the coral health at the time of sampling. And after they were sampled, they were frozen in liquid nitrogen to preserve that physiological state to have the most accurate representation of the coral health status at the time of sampling. So for the extraction methodology for the trophic states, essentially we ground up the corals, the frozen corals, and we applied an NaOH buffer solution that was then analyzed for the protein content using the Bradford assay developed by Bradford in 1975. For the skeletal surface measurement and the symbiote biomass, the symbiote biomass was conducted using a zooxanthellae cell count. And for the skeletal surface measurement, we just measured it in squared centimeters. And both of these methodologies were actually created by my supervisors, Tamala Zellman and Paula Furla. For the biomarkers, it's important to note that for my specific study, we did not include the ubiquitination. If you have questions, I'd be happy to answer them. But we did include the total oxyradical scavenging capacity and an ELISA test to determine the amount of carbonylation present. So for the TOSC, it was developed from a methodology from Naguib in 2000. And for the ELISA, it was developed from Buss et al., or Buss and colleagues, in 1997. So just for time's sake, I'm just going to show you the results of Poecilopora mandrina. If you have questions later, I'd be happy to answer. So from our first trophic state measurement, we have the animal biomass. So from the analysis that was conducted, we can identify that there was no significant difference between temperate and tropical locations. However, there was a significant gradient between Sissoko and Fiji. And that is represented by the letters on top of the graph as it correlates to significance similarities between the islands, not the temperate and tropical locations. For the symbiote biomass, we observed no significant difference between temperate and tropical locations and no significance between the islands. Regarding the carbonylation and TOSC, we also observed no significant difference between temperate and tropical locations. However, we did observe some very interesting trends. As we can see in Sissoko, for both signature markers, we notice that they're very low in comparison to the rest. And while they're very low, because both of them are very low, we have a low carbonyl and a low TOSC. That means that the coral at the time of sampling was not exhibiting an oxidative stress. However, let's say if the Sissoko is low here and we have a much higher TOSC response, it could be indicative that the corals may be fighting something at the time of sampling. If the carbonyls are extremely high and the TOSCs are very low, that indicates that the corals are subjected to damage as the TOSC is the antioxidant response and it's diminished. So as we can see as well, all of the islands had something in common except for Sissoko. It was significantly different when compared to other islands. So regarding our discussion, for Poecilopora mandrina, it exhibits higher contrast between the islands. This is in relation and in accordance with the results that were observed from a paper published regarding the 11 islands within the Terra Pacific expedition previously analyzed. And what this essentially means is that Placillopora mandrina is known for exhibiting phenotypic plasticity based on its environment. And we could see that with the variation in the high contrast in our different biomarkers. However, for the Great Barrier Reef, we did observe low animal biomass in every single, in animal biomass, but in the trophic states as well. And so we have to think about the considerations that I'll get into a little bit more that would cover more of the anthropogenic pressures, but it is a total takeaway of what we observed. Regarding Sissoko Island, the low oxidative states, this was also observed in Porritas lobata, which is very interesting as we have the two resulting responses from two varying species. So Porritas lobata is less flexible, but is less strongly represented between the islands. What this means is that Porritas lobata exhibits lower phenotypic plasticity and is essentially a robust phenotype, meaning that there was observed less variation between the biomarkers. And this was also what was seen in the resulting 11 islands. So for Sissoko Island, a consideration that we can consider for both species, and in addition, the Porritas lobata exhibited the same results in that we rejected our initial hypothesis of temperate versus tropical. However, for Sissoko, there's a paper that was recently published regarding the reef status in these islands, and that they recently undergo a mass bleaching event that they're moreover recovering from. So we can make indications about that. Though the low oxidative state may be linked to metabolism, that is a broad assumption. The additional Terra data that is to be realized will help us make inferences about the possible low oxidative states in Sissoko. Once the global results are produced, we can make inferences based on the contextual and physical data. For the Great Barrier Reef, it's important to consider the possible higher amounts of anthropogenic stressors, the lack of variation in temperature, which could ultimately amount to less food availability and nutrients in the environment, and more pollutant runoffs. Ultimately, the temperate and tropical hypothesis must be rejected. However, we need to account for scalability within the size of this specific study, and that we only included 4 islands rather than the 32 archipelagos that are involved in the Terra Pacific expedition as a whole. So to conclude, there are no significant differences in the temperate versus tropical reefs. However, there is additional data that is yet to be realized by the Terra Pacific expedition that includes the genetic analysis of the coral sample that could give us more inferences about the genotype to phenotype relation and the expression of phenotypes in various environments. Ultimately, the Terra Pacific expedition is still the largest coral sampling effort to give better indications of our coral health status today, but also to create inferences about where our coral reefs will be tomorrow. Thank you.
Okay, guys, you can start if you have any questions.
Yes. So well done, that's a very good presentation.
Thank you.
If we couldn't say that there was a difference between the temperate and the more southern islands, would it be more than species specific how they handle the changes within their location?
So that's what's very interesting about the other results is that just to be very clear, this analysis is not between species, as we can't do that. They have different known physiologies, so they're not comparable. However, sorry for those online, but what was really interesting is for Porritas lobata, when we would see the results of the 4 islands, they were very, very similar and not a lot of high contrast. When we have the Placillopora mandrina, especially for the trophic states, we observe high contrasting variants, which is super interesting because we know from the previous results of the 11 islands that the plasticity response within this specific species is always going to be higher based on the environment. So I thought that was very, very interesting as it was in concurrence with the previous results, but it was also really well represented in even just this small study. even though we didn't find significant differences with the statistical analysis of the temperate versus tropical, in the sampling size, I think would be really nice. Like once we increase and we have the global Terra data, most likely there will be more indications of how they're managing in these environments. And also the Poritas lobata and the Poecilopora mandrina, what this expresses based on the contrast in their biomarker response is 2 responses to the rapid changing environment. So it's also interesting to see how specific organisms will respond to climate change and see the threshold of their adaptive abilities over time.
Thank you, Calli, for your presentation. Very interesting. So I have several questions. First of all, can you remember me what is the sampling size for each site?
Yeah. So every single island except for the Great Barrier Reef had 3 sampling sites. The Great Barrier Reef had 4. So for each site, we had 5 colony samples, and then per site, per island, per species. So essentially, when we were done, and everything was resuspended. We had about 130 protein samples. But for the coral itself, it was just the 5 colonies per site. So yeah.
Okay, thank you very much.
Mm-hmm. And only unbleached. Only non-bleached corals were sampled.
And another question, you mentioned that Tara Ocean was, the whole expedition was during 1 year?
I'm sorry, it was 2 years.
2 years. So do you think that there can be some factors as seasonality that can affect also your results?
Yeah.
Your sampling done in the same period?
Yeah, for sure. So what's interesting is that, if I go back, actually, I shouldn't have left. Okay, so Ogasara was in the northern hemisphere. It was during winter in February, and this was, winter, spring. And the same for Sissoko was sampled in April. For Fiji, these were also sampled during, for the Great Barrier Reef, it was winter for sure. These were June, and these were sampled from August to September, so like on the outskirts of winter, which is super interesting for the discussion and the consideration of the trophic state of those environments as well, because one thing to consider is that when we don't have a thermocline mixing, the nutrients available in the water decreases. And also if we think of things like cloud cover and other environmental factors that may influence the success of photosynthesis at that time, it can also directly relate to the animal biomass that's presented. So the seasonality is very interesting as well. I believe there's another Terra Pacific expedition that they want to do in 2025. Don't quote me, but I believe there's another one in 2025 that they want to do to compare this data and make other samples so we can have a progression of the data. But yeah, that is something that is a factor. While we can produce really nice replicates based on the species and the methodology, as the methodology is standard for every single Terra Pacific analysis, there's like 23 research institutions that work globally to produce these results. There is variability in the sampling time, so that's why I was like temporal, because it does give indications on the health status and the variation that may be seen within the trophic and the oxidative states at the time.
And based on your knowledge, how do you expect that another sampling in another season can affect the result, particularly for the oxidative stress?
Mm-hmm.
What is the mechanism that can affect it and how?
So for oxidative stress, there's many different parameters that could negatively influence it, because our cells naturally produce the reactive oxidative species, and the problem is when they're produced in excess. So some of the parameters that influence this can be temperature, salinity, different states of the environment. So what I think would be really interesting is sampling, like if we could have more contrast between, if we're talking about a temperate and tropical consideration, it could be interesting to kind of sample these guys like dead of winter and these in the heat of summer, because then we're going to show also completely opposite nutrient availability. And for oxidative states as well, the temperature increase is known to reduce the enzyme creation or like the enzyme production for antioxidants. So for corals, it's known that when we increase the temperature, we also can reduce the amount of antioxidants available to fight external stressors, which ultimately leads to a higher amount of reactive oxygen species within the cell.
Okay, yeah, perhaps I have just one question, because like you said, you focused on only one species. Do you have, like, just briefly, can you, I know at the end you said there was like no significant difference, but did you find something perhaps
More interesting?
Yeah, more interesting in the second species?
What was interesting for me personally was just to be able to really visualize, because like it was really like trophic state, oxidative states were very similar in the fact that we could see that the phenotype was staying very similar, which was really nice. But ultimately, what was very interesting about this particular study was Sissoko, because they both exhibited extremely low oxidative states, which, as I said, it doesn't mean that they're in stress. It actually means that they're experiencing, I don't want to say no stress because it's not possible, but they're experiencing abnormally low amounts of stress. And so that's why Paula actually sent me a paper about the research that was done, is because we're kind of thinking like, okay, what's going on there? Like, is there an MPA? Like, were there new ordinances that were put in place? Are we replanting? Is something going on after this mass bleaching event? But also the Great Barrier Reef, and this is for Porritas lobata, sorry, but this is Postilopora, but the results were very comparable. For the Great Barrier Reef, we also noticed in the Porritas lobata that we had a low animal biomass represented in the Great Barrier Reef, which got me to thinking more like biological context as to maybe it's because of anthropogenic stressors. We know, like with sunscreens and everything, the effect that pollutants can have on the coral reefs, but ultimately there's something going on with the photosynthesis that's not allowing the proper nutrients to support the animal tissues growing. But essentially for Porritas, we had the same results regarding the temperate and tropical, and we also had significant similarities between the islands. Very similar results to Postilopora and Mandrina. I just didn't include them for time's sake.
Yeah, I understand, but you don't have the graph, so the graph are looking quite the same in the idea that you have the same trend in the other species?
No, when I said what they look the same, so we can see this high contrast when compared to Porritas. Sorry for those of you that are online, essentially. The graph was pretty linear for each of the islands. So what that means, based on the literature, is that it's exhibiting that there's not a lot of phenotypic plasticity within the species. While when we have Postilopora mandrina, we see high contrast between the species, which I think is very interesting because it was a very obvious result. So for most of, I'm sorry, I didn't put them in the appendix, but for most of the Porritas, we see really similar trends for the trophic states and for the oxidative states.
Okay. And perhaps just like briefly, last question for me, it's just like, because so it's been a project, like you said, it's a long time, so there is also students who went like for Pacific before you. So what did you continue from the previous students, and what is going to be the next step, really? So pretty much what the next step is for, I didn't really continue anything other than the analysis. Okay. So I know Mariana was before me, and she did an island-specific analysis between different island sets within one area. My scope was a bit different based on temperate versus tropic. The idea is, I don't want to say like one of the negative things, but one of the things that needs to be understood is these results take a lot of time for a realization. And so these sampling efforts for my islands occurred in 2017. So the next intern that will be there will continue the same process, but just to tick off the islands so we can realize those global results. And Kayla is also working, Kayla Plachon is also working with Paula now to further help speed up the process
of realizing the results because the data is very significant. However, we just need to dig it up.
So you, like you said, there was like so many different things were analyzed. So from all the results you have until now, what are like the main kind of conclusion you can say? Perhaps it's going like further than your subject, but if you talk with Paola, with the other students, I guess you have a global picture of it.
So what like I do and what I do under supervision of Paola and Tamala is just the biomarkers. I'm not involved in the genetics in any way. That's like another team. There's a whole like genetics team, but Didier Foselli is part of that team as well, and he's explained some of the results from the papers. But it's more so attributing to how the species in and of itself will respond in the environment. So what was nice about the result, and I'm sorry, this is repetitive, but it's mostly just the results that were produced in the 11 previous islands. We're stating that, okay, for this species, we see this in this environmental context. For this species, we see this in this environmental context, which I think is super cool because while I was expecting to see more dramatic results, because I'm like, okay, we have tropical, there's no nutrients, high temperatures, they're gonna die. Like I was like, this is problematic. However, we see corals in that state, and I mean, even in 2017, we see increases of temperature that are still able to withstand the environment to maintain a healthy state, meaning they can produce a reproduction cycle. Kayla.
Okay. Yes.
Kind of a follow up on that. Not really. Anyway, how many labs are involved in this project? Because
23.
Is it just France?
No, it's like institutions globally, and so they like ship the corals in nitrogen to different labs that are participating with the Terra Foundation, and it's like it's a huge collaborative effort, which is also I think is super cool, given the context of the global environment and what's going on. It not only produces globally relevant results that are important to the scientific and just general society, but it's a huge collaborative effort between many different people, which I think is really, really cool. And I'm so happy I got to be a part of it.
I can imagine. It's probably hard to answer, but do you have an idea of how many samples were analyzed? Because it must be a huge, like.
For mine, it was like 130.
Like in percent, like how far are we in the whole analysis?
It's gonna be done soon.
Oh,
like
really?
Probably like 1 year. I think I don't quote me, but I think that we're at the end of the analysis for the genetic analysis. I think it's like between like 11 and 15. I don't know. Don't quote me. I'm sorry.
It's fine. I was just curious.
Yeah, but the idea is like with Kayla, she'll produce a lot of results to be realized, and then the next Terra Pacific intern will help with just getting the data out there.
Well, Paola, if you want to add something.
So thank you very much, Calli. And to answer the question, yes, it's a so long project. And I hope that all the phenotypic data that Calli helped to obtain will be finished next year. But there will be next some temporal analysis, and I understand that 2 new corals will arrive soon, so it's never finished, the Terra Pacific project. So I would like just to add that probably you know even better than me, Calli, but Calli was very lovely student, very attentive and strong-willing, and she's also very talk active, as you may notice, and it was really a pleasure to work with her. She was very involved in her work. She has also some issue with one of the markers that she didn't show you. She explained ubiquitin, but she would not be able to obtain result because we have at that time many issues. She spent quite 2 months to try to resolve this, and Tamina is still trying to have some better results. So it was really complicated for her, and that is also it's not easy for a student to appraise this project because there is no control conditions and stress conditions or treatment conditions. There are only natural islands, natural population, of course, and so it's always tricky to compare. And the best comparison that will be done will be at the end of the project when we will have all the 32 islands. So just to finish with Calli, and I hope it will be the same for our corals. Calli also had the ability to acclimatize so well to a team, and she left an excellent memory to all of us. So many thanks, Calli.
Thank you, Paula. I just say that. Wow. Okay, but I just want to say a big thank you to everyone on the Eric Radinger team. It was truly a pleasure, and to me, it is truly a seventh heaven. So thank you so much. Thank
Yeah,
you, Paula.
we can also thanks very much Kayla that
Yeah.
helped you a lot also
Yes.
these last days. So many thanks.
Yes, Kayla was helping me prepare my presentation and was my coach. So thank you very much, Kayla. I hope you liked the reproduction I put for you.
Okay, so thank you guys. Thank you,
Thank you,
Paula.
Paula.
So we will be back at 1:30 for this afternoon presentation. Thank you to be online, and if you want to follow us, we'll be at 1:30. Bye.